Hi, in this video, we are going to understand the ninth chapter, that is, Heredity and Evolution, which is divided into two parts. In this video, we are going to understand the first part. We will cover the following topics in this video. First, we will know what is heredity and variation. After that, we will know about types of variation. After that, we will learn about accumulation of variation during reproduction. We will also study about important terms. The video mainly helps in study about heredity, that is Mendel and his work on inheritance with their experiments in detail. So, let's start with our first topic. The very first thing that comes to our mind before starting the chapter is, what is heredity and variation? As we know that, genetics deal with the study of heredity and variation. The transmission of characters or traits from one generation to the next generation is called heredity. The differences in the characters or traits between the parent and offspring is called variation. Variation is caused by different combination of genetic material, some positive gene mutations, and interaction of gene with environmental changes, that is adaptation. There are several importance of variation. They are: variation forms the basis of heredity. It helps in adaptation. Types of variation. Variation are of two types. They are: first one is somatic variation or acquired traits. It takes place in the body cell. It is neither inherited nor transmitted. Examples are cutting of tails in dogs. Second one is gametic variation or inherited trait. It takes place in the gamete or reproductive cells. Inherited as well as transmitted. Examples are human height, skin color. Now, we will study about accumulation of variation during reproduction. Variation occurs during reproduction, whether organisms multiply asexually or sexually. Let's study about it. Variations in asexual reproduction. In asexual reproduction, like bacteria, the parent will give rise to two individuals similar in body design, but with subtle difference each of them, in turn, will give rise to two individuals in the next generation. Results are, variations are fewer and occur due to small inaccuracies in DNA copying. Variations in sexual reproduction. For this, Mendel took pea plants with different characteristics, a tall plant denoted by capital TT and a short plant denoted by small tt. They are parents and crossed them. Produced progeny have all tall offspring denoted by capital T and small t. They are F1 generation. Now, these F1 tall plants, to be produced by self-pollination, gives not all tall plants. Instead, 1/4 of them are short. It results larger variation, and these variations occurs due to crossing over and separation of chromosomes and mutation. Before starting Mendel's work, we should familiar with some important terms, which is helpful in study further. They are gene, basic unit of inheritance, characters transferred from parents to their offspring. Genotype, complete set of genes present in an organism. Phenotype, observable character in an organism. Dominant trait, allele, affects phenotype of organism. Heterozygous and homozygous condition denoted by capital letter, that is, tallness is denoted by capital T. Recessive trait. Allele affects phenotype of organism in absence of a dominant allele, that is homozygous recessive, denoted by small letter. Dwarfness is denoted by small t. Pure or homozygous condition. The condition in which both trait or alleles are dominant, that is capital TT, and both traits or alleles are recessive, that is small tt. Heterozygous condition. Hybrid. The condition in which one trait is dominant and one trait is recessive, denoted by capital T and small t. Our next topic is Mendel and his work on inheritance. Gregor John Mendel was known as father of genetics. Plant selected by Mendel. Pisum sativum, garden pea. A question come in mind, why garden pea? It is because they are easy to grow due to short lifespan of plants, easily distinguishable characters, normally allows self-fertilization, but cross-fertilization can also be carried out, large number of seeds produced, and so on. Lastly, we will talk about 7 pairs of contrasting characters in garden pea. The contrasting characters traits are height of plant, seed shape, seed color, pod shape, pod coat color, and flower position. The dominant traits for the characters are tall, round, yellow, green, inflated, green, and axial respectively, while the recessive traits are dwarf or short, wrinkled, green, yellow, constricted, white, and terminal as in the chart. Mendel's experiments. Monohybrid cross. Cross between two pea plants with one pair of contrasting characters is called a monohybrid cross. For example, cross between a tall and a dwarf plant. He took purebred parental generation, a pure tall plant, that is capital TT, crossed with a pure short plant, that is small tt, produced progeny is F1 generation. There are no medium height plant. All plants were tall offspring, denoted by capital T and small t. F1 plants were left to self-pollinate, gives F2 generation. He observed that all F1 progeny were tall, no medium height plant, F2 progeny, 3 by 4 were tall and 1/4 were short. Phenotypic ratio of F2 progeny were 3:1, 3 tall is to 1 short, and genotypic ratio were 1:2:1, 1 pure tall, 2 hybrid tall, 1 short plants. Through monohybrid cross experiments, Mendel proposed 2 laws. First one is laws of dominance. It states that one factor in a pair of traits dominates the other in inheritance unless both factors in the pair are recessive. And the second one is law of segregation. It states that during production of gametes, 2 copies of each hereditary factor segregate so that offspring acquire 1 factor from each parent. Now, the next topic is dihybrid cross, a cross in which 2 pairs of character are studied is called dihybrid cross. For example, cross between 2 pea plants, one have round yellow seed, other have wrinkled green seed. Let's study about it. He took purebred parental generation, a pure round yellow seeds, denoted by capital SSYY, were crossed with a pure wrinkle, green seeds, denoted by small ssyy. All the F1 generation showed only the dominant form for both traits. All hybrid, that is, all yellow and round seeds. When these F1 generation allowed to self-pollinate, F2 seeds were obtained. Their phenotype ratio was about 9:3:3:1 ratio. At last, through dihybrid cross experiments, Mendel proposed third law, that is law of independent assortment. It states that alleles of different characters separate independent from each other during gamete formation. Now, the next topic is sex determination. Sex determination is the process of determination of sex of an offspring. There are several factors which is responsible for sex determination. They are, first one is environmental factor and second one is genetic factor. Now, at last we are going to study about sex determination in human beings. In human beings, 23 pair of chromosomes present. Out of these, 22 pairs chromosomes called autosomes and 1 pair called the sex chromosomes. All human chromosomes are not paired. Women have a perfect pair of sex chromosomes, both called X. Men have a mismatched pair in which one is normal sized X while the other is a short one called Y. When both parental gametes, sperm, that is paternal gamete, and egg, that is maternal gamete, fertilized and produces offspring as shown in the chart. The chart shows half the children will be boys and half will be girls. All children will inherit an X chromosome from their mother regardless of whether they are boy or girls. Therefore, sex of the offspring in human and in mammals is determined by what they inherit from their father. So that's all in this chapter. Thanks for watching.